Okay, what we want to look at here is this lipid and protein catabolism. What we looked at in that previous section, very long section there, was carbohydrate catabolism, or that is the respiration of carbohydrates, breaking down carbohydrates such as glucose down to carbon dioxide and water, and transferring that energy from the carbohydrate molecule into ATP. The ATP can then be used to do work in the cells. That applies whether we're talking about bacteria, human, plants, fungi, or what have you. Now, carbohydrates pretty much across the spectrum of life. Those carbohydrates are going to be the main source of energy, and the main carbohydrate that is going to provide energy pretty much across the whole spectrum of life is going to be glucose. Okay? But the main gist of this section right here is that other compounds, other organic compounds, such as lipids and proteins, can also be broken down, or that is catabolized, and their energy transferred into ATP. Okay, so let's go down here and find a few things. Right here, we have the term lipase. Okay, lipase is going to break down fats into their monomers. Their monomers are going to be fatty acids and glycerol. So, there we go. So, the lipases are going to break the fats down into fatty acid and glycerol. Notice right there the word extracellular. So, that means rather than happening inside the cells, this happens outside the cells. So, instead of, in our case, where we have digestion within the organism, these microorganisms, specifically bacteria and fungi, are going to secrete these enzymes, the lipases, to the outside of the cell. The large complex organic molecule will break down to its monomers. In this case, fat breaks down to fatty acids and glycerol. And then those components are absorbed into the microbial cell. Now, once in the microbial cell, we're going to have Krebs cycle. It's going to act on these. It's going to hydrolyze the fatty acids. And they point out that the same enzymes that can break down fatty acids can also break down petroleum products, which, depending on the situation, that can be a benefit or a problem. Here we go. So let's take a look. Lipid catabolism, in other words, breakdown of lipids. So we've got fats up here. Now this ought to have an arrow. Whoops, draw on that. This should have an arrow going over here as well, because that fat, which is going to consist of glycerol and then 3 fatty acids. So basically right there, glycerol and then the fatty acids right there, those are going to be cleaved apart by this enzyme lipase. Okay? So the enzyme lipase will cleave that apart. Now glycerol is going to feed basically into the middle of glycolysis, and it's going to produce pyruvic acid, which will then produce acetyl coenzyme A, which is going to be this coenzyme A right here and these 2 carbon units. Okay, and that'll feed into Krebs cycle, and then from Krebs cycle on into the electron transport chain. The fatty acids are broken down into 2 carbon units, and then we're going to have this enzyme CoA attached to that, which will be able to carry that into the Krebs cycle then. Okay, so basically, also right here, since the glycerol is a 3 carbon molecule, we'll have one of those carbons given off as carbon dioxide right there. Okay, another thing to realize about this is we go up here and look at this fatty acid consists of this carbon chain, like a fairly short carbon chain right here, and we have hydrogens all along this carbon chain. So if we go back to what we looked at for carbohydrate, the breakdown of carbohydrates to transfer that energy to ATP, we remember that the carbons and the oxygen were essentially garbage. Those came off as carbon dioxide. So basically right here, our carbon, that's going to go off just as carbon dioxide. Here's the part we're going to capture and use. That's an H, not a 4. Here's what we're going to capture and use. So notice what you have. This fatty acid right here is just loaded with hydrogens. So basically right here, this fatty acid has a high number of hydrogens. Thus, this fatty acid is very high in energy. Okay. Now, because of this, we have about 4 kilocalories per gram for carbohydrates, and by comparison, we have about 9 kilocalories per gram per gallon. That was supposed to be per gram in the fats and oils, that is the lipids. So we've got a lot more energy in these lipids than what we do in the carbohydrates. Okay, let's see what this next one is right here. Okay, this next one is looking at the use of proteins, carbohydrates, and lipids in this process of aerobic respiration. So where we started was with carbohydrates. Carbohydrates are the sugars, and it's going to go in right here at the very beginning of glycolysis, go all the way through the process of glycolysis, through the Krebs cycle, and through the electron transport chain. And if you notice the lipids up here, they will enter right here and right here. Okay, the proteins are going to be broken down into their amino acids, that is their monomers or building blocks, and depending on which amino acid it is, it can enter here as pyruvic acid, it can enter here as acetyl coenzyme A, or it can enter somewhere in the Krebs cycle. Okay? But the point is, all of these compounds, proteins, carbohydrates, and lipids, can be used to transfer their energy ultimately into ATP by going through this process of aerobic respiration and ending up down here with the electron transport chain and chemiosmosis. Okay, so let's go back over the textbook and look at that. And let's go down here and look. Now, one of the things they point out, just like up here with the fats, let's go back up and find that statement with the fats here. Our lipase is an extracellular enzyme. So it's going to secrete that enzyme, lipase, to the outside of the cell. The chemical reaction of breaking the fat down to its components will take place, that is, the digestion of fat will take place outside the bacterial or fungal cell. Then that microbial cell will absorb those broken down components. Well, let's go down to proteins. And right here, proteins are going to be too large to pass through that plasma membrane. And remember, in animals, we have endocytosis. We have a mechanism to bring large particles into the cell by that plasma membrane folding inward and pulling those large particles in. These bacteria, as well as fungi, but let's stay with bacteria. Bacteria are surrounded by a cell wall, and so they're not going to have direct contact with these proteins that are on the outside of the cell. So they don't carry out this endocytosis. where this plasma membrane folds inward and pulls large particles into the cell. So they've got to have a different way of getting these things like a large protein into the cell. So notice what's going to happen. They're going to produce, there's that word again, extracellular proteases and peptidases. Proteases are going to break down proteins. Peptidases are going to break down peptide chains. They're going to break that down to their individual amino acids. The individual amino acids now, the monomers, are small enough that they can go across that membrane and get into that bacterial cell. Okay? Now then, remember that amino acid. Let me go back over here and sketch out an amino acid for just a second. Remember this amino acid right here. Here's our amino acid. You have, whoop, let's try this. Come on, come on, come on. Trying to, here we go. Now maybe I can draw. Okay, we've got a central carbon. We've got a carboxyl group on one end. We have our R group. That's the R group is what's different between the 20 different amino acids. We have a hydrogen. And then right here on this amino acid, we have a nitrogen and 2 hydrogens. That is the amino. That is the amino group right there. Okay? This is the carboxyl group right down here on this end of the amino acid. And of course, this whole thing is going to be an amino acid. Okay, remember what happened in processing of sugar, what happened in processing of fat. Basically, we took this 2-carbon chain right here, such as this 2-carbon chain, and we fed that into Krebs cycle. Okay. Put that into the Krebs cycle or citric acid cycle. This nitrogen group really does not have a place in that set of series of chemical reactions in that metabolic pathway. So one of the things that has to happen is we've got to get rid of that nitrogen group before this carbon chain can feed into respiration. Okay? That process, since it's called an amino group right here, this process is called deamination. Okay? Deamination. Removal. That is a removal of the amino group. That has to happen before this amino acid can move into the process of respiration. So notice right there is that word deamination. The amino group has to be removed from the amino acid, and then it's going to pick up additional hydrogens and form that NH4 with a plus charge on it. That's an ammonium ion. That's going to be given off from the cell. Okay. Now, the other group that we had in there was a carboxyl group. So we're also going to get decarboxylation, removal of the carboxyl group. And if we have one of those R groups that contains sulfur, we're going to get desulfurization. That sulfur group, here's our central carbon, here's our R group. Some of these R groups will contain that sulfur. Okay? So, not all of them, but some will contain that sulfur. Okay, here's the diagram that we looked at just a moment ago. So let's go on down here, and let's go over to this next section right here. So, we've got the idea that these bacteria can essentially digest carbohydrates, they can digest lipids, they can digest proteins. Well, the way that they digest these and the different end products that they produce can be used to identify the bacteria. You've looked at enough bacteria in lab by now that you know a lot of these bacteria look alike under the microscope. So let's go down here, and we have these biochemical tests that will detect different types of enzymes that cause decarboxylation, dehydrogenation, such as this. And so detecting amino acid catabolizing enzymes, and basically right there you get a color change, and down here it describes the one that turned yellow is where the bacteria produced an acid from glucose. The other one had process of decarboxylation, and it produced a alkaline solution. So both of those are based on pH changes, but those pH changes are going to be reflective of different metabolic pathways occurring within each of those tubes, which would allow us to identify or at least narrow down what the bacteria might be. Okay. Here's another example, and this is a fermentation test. And so with the fermentation test, we're going to have a protein, one single carbohydrate, it might be glucose, it might be maltose, some other carbohydrate, a pH indicator that can tell us if it formed an acid, and what's called a Durham tube, which is just essentially a small test tube to capture gas. Okay, so let's go down here and look at this, and here's a fermentation test, and on the first one, A, there was no color change. It's still, notice down here it says A, uninoculated fermentation tube containing carbohydrate mannitol. So that's essentially our control, is A. Okay, on B, there was no color change, but, and it's kind of hard to see in the photo, you have gas produced in that small test tube. Okay, so what that tells us is Staphylococcus epidermidis can grow on the protein, but it's not using the carbohydrate. If it had used the carbohydrate, it would have produced an acid, and we would have gotten a color change. Okay. Now then, on C, you get the color change indicating that an acid has been produced, but in the small tube, it's still liquid. You don't have gas produced in there. So, Staphylococcus aureus is going to produce acid, but it's not going to produce any gas. And notice the species that cannot use mannitol, we refer to that as mannitol negative. Species that can use mannitol, we call that mannitol positive. Now, the last one was inoculated with Escherichia coli, and it was able to use the mannitol, change the mannitol to an acid. You know that because of the change in the color change in the pH indicator, and kind of hard to see, but the small tube has gas in it. So it produced gas. So Escherichia coli is also mannitol positive. It produced an acid and it produced gas from mannitol. And so this allows separation of these different species based on biochemical test. Okay. Now, here's another one. E. coli will ferment the carbohydrate sorbitol. But the pathogenic strain of E. coli does not ferment sorbitol. So there's a biochemical test that can be used to distinguish two bacteria of the same species that are going to look just alike under the microscope. But this biochemical test can differentiate those and tell the pathogenic version from the non-pathogenic version. Let's see. Down here, they give you some other examples of biochemical tests. Biochemical tests are used to identify bacteria that cause disease. And one of the things it points out, all aerobic bacteria are going to have an electron transport chain, but those electron transport chains can differ from one type of bacteria to another. So we're going to do biochemical tests to identify those. And here they talk about a biochemical test to identify Neisseria, gonorrhea, and likewise Pseudomonas and Escherichia, Shigella, and so forth. Basically, based on what types of, if I can get it to come back down here, notice, for example, right here, it's trying to come back down, based on, in this one, whether it will utilize lactose or not, which we determine by its production of gas. Okay? Production of hydrogen sulfide could be another distinguishing factor. So Salmonella, readily distinguished from E. coli by their production of hydrogen sulfide. And we'll go down here. The way that's going to work is this hydrogen sulfide will react with a peptone iron auger, which will cause the precipitation of iron in that auger. Okay? And so that can tell us that, oh, this was Salmonella rather than E. coli. So it's just these biochemical tests can be used to identify the bacteria, and that's taking advantage of all the diversity in metabolic pathways that we have in these microorganisms. Okay, so let's stop this video right here, and we'll pick up with one, hopefully just one more after this. Okay.